came from Kotzebue. So we're really glad he came down. He's had a very unusual day as well, being sort of watching all this unfold, but not being home. So with no further ado, I do want to mention that not only is Franz a professor, but he also serves on the North Pacific Fisheries Management Council in their science and statistics.
Scientific and statistical committee.
Yeah. So that's know that there's questions you can ask him at that level as well. And with that, take it away, Franz. I'm going to hit these lights.
Thank you.
We're so glad you're here.
It's great to, yeah, you can turn that one off, but yeah. Yeah, it's great to be back. I always love coming to Nome, visiting. It's kind of short now, I hope at least. I hope I'm going to try the weather window tomorrow to get out before they close the airports too. So yeah, I was just going to kind of tell you a little bit about the cruise, but a lot of it is just sort of a little bit of background on Arctic Cod, which is a species that we were interested in. And so we did set up in November, and the pictures of everybody on the boat, the names here are just the people that were involved in the sort of the biological Arctic Cod project that we were interested in, and there was a whole other crew, a different project, the CODA project that Jim Thompson, the chief scientist, led from the University of Washington on looking at the impacts of waves on shore-fast ice in particular, but ice in general. And so our main goal for the cruise was really to just look at, well, really two goals. For me, one of the main goals was to see if we could use a new piece of equipment that we acquired a few years ago. That's an under ice trawl. If we could actually use that and deploy that off the Sikuliaq. It was the first time that's been used in the Pacific Arctic, and they deployed off a very different vessel in the European Arctic off the Pelagstein, the German icebreaker that's frozen in the ice in the Central Arctic Ocean right now. And so that was my big goal, just to see if we can make that work. And then scientifically, we were interested in how Arctic cod associate with ice in late fall. You know, we don't really know much about them at that time of year, and I'll kind of give you a little bit of background on that and why we were interested in that. So this is the Sikuliaq. You can actually see the net here, and you'll get a better idea of how that works in the video later, although it is kind of hard to capture that in images. You almost have to kind of be there to see it. But you can kind of see the net trawling behind here. It's a frame here. It's a big frame, 6 by 6, 7 by 7 foot roughly. And then it's got floats on top. You can see these floats on top. I should use a pointer so Bob and Vera can see that too. So it's got these floats on top here that keeps it at the surface. And then there's a cable connecting it to the boat, going through a ship on the A-frame here. And there's a heavy weight attached to the cable that pulls the cable down, so you get the cable under the ice. And then when the ship steams along, the water flowing into the net here basically makes the net shear out to the side. So it actually tows about 60 to 100 feet to the side of the boat once you get about 100-meter wire out. And you'll get a better idea of that later when I show the video. But before I go to the video, I want to give you a little bit of background on the star of the show here, Arctic cod, who love ice. And when they ice like this, they're a little hard to sample, but we were interested in seeing whether they actually associate with the young ice, like the ice that forms first in the fall. And so I'll tell you a little bit about what we know about them and why we're interested in them. So they are a pretty key component in the Arctic food web, connecting both the ice algae and phytoplankton in open water via zooplankton, primarily copepods, like calanoid copepods, these small zooplankton, to their predators, and that includes seals, belugas, seabirds. The seabirds mostly feed on the young ones in the water column, and then ultimately, polar bears and humans. So they really play a pretty central role in the Arctic food web that way. As an example, about 49 to 94% of ring seal stomachs and 50% of bearded seal stomachs in that period here contained Arctic cod, so it's a pretty important part of their diet. They distributed all over the Arctic. That kind of roughly matches the maximum extent of sea ice and a little bit beyond it in you know, on the east coast, off the east coast of Canada. They like to be either on the bottom or in the water column, often associated with sea ice, but they can kind of be anywhere in the water column, really. They typically mature about 2 years, some even at 1 year, 2 to 3 years more typically. Don't get very big, 25 inches, mostly smaller than that, particularly in the Pacific Arctic. Typically no older than about 5 years old. A lot of them die after spawning once. They can get up to about 8 years. And they're really well adapted to living in these conditions. They have kind of big eyes to see in dark conditions because they're visual predators, and they have antifreeze in their blood, so they can withstand freezing temperatures. And they grow pretty well in very cold conditions, much faster than other fish that aren't adapted to those cold conditions. I first want to show you a little bit about what's happening kind of on the edges of their distribution. I had a student who graduated recently looking at 2 of these regions, well, a couple more actually, but I'm only going to show results from the Bering Sea here. But we know most about them in these regions because that's where fishery surveys are happening, including the survey in the Bering Sea here that you know, you guys are probably familiar with. So there we actually have a time series. We can actually say something about how their distribution has changed over time, and that time series spans 82 through 2019. The northern Bering Sea, of course, was only sampled a few times, so that's not included here. And so for the analyses, we basically just looked at this blue wedge here because they never occurred throughout that time period. They never occurred to the south of that. And so Jen just plotted out the bottom temperatures each year and the southern extent of the Arctic cod. So the blue here, or purplish, is the cold pool, so it's water below 2 degrees Celsius. The reddish colors are warmer, obviously. And then the black line is basically the kind of the southern extent of Arctic cod that year. And you can kind of see that their distribution kind of tracks the cold pool pretty well. Yeah.
And so would the cold pool be reflecting that where the ice is located?
The cold pool reflects where the maximum extent of ice was the winter before. So that's typically, you know, when the ice forms, the whole water column gets cold top to bottom. And then when the ice melts, then the bottom layer will typically stay cold, even if as the surface warms up. It takes a while for that bottom layer to erode, either from the sides or from the surface. And so typically you get the cold pool all summer long. It stays there all summer long. These surveys are done in the summer, so that's what, it's kind of a snapshot in the summer, the distribution in the summer. But you can kind of see how those that track the cold pool, and I can plot that out too. This is just plotting the fraction of the area in that survey area that's occupied by Arctic cod, plotted against the bottom temperature, and that's just a proxy for the cold pool that just measures sort of the extent of the cold pool. So it's a pretty close relationship. In some years, 2010, 2016, the distribution extends beyond the cold pool, and that's mostly, you can see that in 2010 and 2016 here. I'll use the mouse again. In these shallow areas here in particular, the distribution extends into warmer waters, and that's probably because a little bit in 2016 because there aren't not as many predators in those shallow waters. But generally they track each other pretty well. And so what we were interested in was, are those, is that really driven by temperature? Is it temperature that makes the Arctic cod go back and you know, extend south when it's cold, or go back when it's warm? Or is it really, it's not so much temperature, but perhaps predators or competitors that keep them out of those areas because the predators and the competitors don't like those really cold temperatures. So they only move north when the cold pool retreats. And so they may be basically displacing Arctic cod. So we try to kind of untangle that, and that's kind of hard to do, but so this is the most complicated figure I'll have, so I'll kind of walk you through that. But basically, on the left here, the leftmost column, all of those colored dots here just shows the kind of from a statistical model that predicted abundance of, so it's kind of a smoothed abundance of Arctic cod under low temperatures at the top here, under low capelin abundances at the second row, under low Pacific cod abundances, and low walleye pollock abundances. Oh, yeah, sorry. So I have to look back at the arrow because the arrow isn't here. So this left-hand column, that's under low temperatures, low capelin, low Pacific cod, low walleye pollock, and the same under high temperatures, high capelin abundances, high Pacific cod abundances, and high walleye pollock. How do the abundance of Arctic cod respond to those? And then this is just the difference, kind of the change from low to high. And so what this says, these are all negative. Blue is all negative, red is all positive in all of these. So whenever it's warm under high temperature conditions, Arctic cod go down. That's what this says. That's, everything's blue here. This one here is capelin, and we had thought of capelin as a competitor. So we thought that if Arctic cod are gone, capelin might kind of occupy that niche. And if there's lots of Arctic cod around, they might keep capelin out, because that's been hypothesized and observed in some other areas. We did not find that at all, because this positive effect here basically says that whenever capelin abundances were high generally, then Arctic cod abundances were high too. So when capelin were doing well or present in a location, Arctic cod tended to be present there as well. So definitely not a competitive exclusion there. And for the predator here, Pacific cod, we were expecting a negative relationship, but we actually saw kind of a mixed picture. In some areas here, kind of in the center of that distribution, there was a negative relationship, so more Pacific cod that depressed the Arctic cod, but not around the edges here. So this red here is both Pacific cod and Arctic cod kind of very in sync. And pollock is actually mostly negative, and pollock we anticipated to be more of a competitor. They don't really prey on Arctic cod. They compete for the same kind of food. So there's some evidence for that whenever there's pollock around, there's fewer Arctic cod around, the blue colors. So hopefully that kind of makes sense. So but essentially the best predictor here was clearly temperature. If you just wanted to predict where Arctic cod are, just using temperature was the best predictor. And basically warmer temperatures, you see a decrease in abundance of Arctic cod. And surprisingly, I mean, we kind of knew that. We still didn't believe it, that it's actually temperature that's driving it. So we tried to look at that in a little more detail. And so this is hopefully not quite as complicated, but basically that shows, again, it's kind of model-based estimates, but the probability that you see Arctic cod at different temperatures. And the solid line here is at all the stations where pollock actually are present as well. So what this says is, in the presence of pollock, if it's really cold, there's Arctic cod around, high probability that you also see Arctic cod. But as soon as it gets a little warmer, above zero, you barely see any Arctic cod if there's pollock around. So when there are pollock around at these warmer temperatures, say maybe, excluding the Arctic cod, competing or chasing them away or whatever. Yeah, go ahead.
Why did it go up again?
probably noise in the data. I mean, these are pretty noisy data, and this is sort of what we call a 95% confidence band for the mean, but there's the actual points would be a lot more variable. And it's also, I think, because it's in the nearshore areas where there's very few pollock around. And so sometimes in the nearshore warmer areas that actually keeps going up if you extend it, they may be able to thrive as well. Yeah.
Does that have anything to temperature? Pollock don't like it cold, so is there some relationship?
Yeah, so they do occur in these cold temperatures. So this is, remember, this is anywhere there's pollock present. So even at these cold temperatures, there are pollock. They can tolerate it, but they can't outcompete Arctic cod. That's what I think is going on. They just, when it's cold, the Arctic cod have the advantage.
Right.
Yeah.
So behaviorally, there might be a difference.
Right.
Older,
Yeah.
like pollock are like crawling on cold.
They just freeze up and don't do anything. Yeah. Like,
Was there a threshold for use for pollock presence? There a lower limit?
pollock were present throughout. I mean, that was actually part of the problem in the analysis, was that pollock were all over the place. There's relatively few stations that don't have pollock, and that's shown here. So before I take that question, I'll point out the difference of this is when there are no pollock present. So then you actually have, regardless of what the temperature is, you have a pretty high probability of Arctic cod being there. So if there are no pollock around, you do get Arctic cod at all of these warmer temperatures. And we know from the Chukchi Sea, the Arctic cod are quite happy even at 10, even at 12 degrees. So I think it's not really temperature that's driving that. It's that the fact that when it warms up, the predators move north and or the competitors and basically exclude the Arctic cod. And you see a very similar pattern for Pacific cod, actually. And so I think there was a question over there. Pacific cod is a predator, and the pattern is very similar for Pacific cod. The problem was even worse in that Pacific cod were everywhere. This is 2017, so we didn't really have much contrast because they were just all over the place. Yeah.
The width of the lighter blue, is that just artistic?
The lighter blue, that's basically, that's what we call a 95% confidence interval. Yeah.
With the minimum and maximum.
Yeah, it's not exactly minimum and maximum, but that's, we think the probability at 4 degrees Celsius when there's no pollock around is about 90% that you'll find Arctic cod there. But we're not certain about that, so it's somewhere between 75 and 95%, something like that. And I'll just skip over this one. This was 2017, and we did the same thing for 2010, which was a colder year. And the patterns are not as strong, but basically the same. There were fewer pollock up north that year, but the story is basically the same. So that was kind of Bering Sea at the southern end of their distribution. And basically, Jen did the analysis through 2017. In 2018 and 2019, Arctic cod disappeared entirely, as you probably know, from the northern Bering Sea because it was so warm. So there were none around, and both pollock and cod were all over the place. So they probably contributed to Arctic cod disappearing. So the thing is, we see these Arctic cod being very sensitive to the temperature changes, but I think it's actually an indirect effect. They are kind of a sentinel species, but what's driving that variability is more the predators or competitors moving north. So what about the Chukchi Sea? We don't have those kinds of time series in the Chukchi Sea, but we do have a couple years of very good coverage from these AMBON surveys. That's Arctic Marine Biodiversity Observing Network. So we have at least 2 years with very good coverage that we compared, and that's mostly further north. So it's one line off of Point Hope, Cape Lisburn, and then a bunch of lines up north. And what we saw there was that in 2017, there were generally more Arctic cod around, and that's the H1, the larger ones on the bottom. So there were more in 2017, which was a warmer year, and fewer in 2015, which was a colder year. Not particularly cold, but relatively colder. Was that a question? And then the other thing that happened in 2017 was that we saw many more species in that region in 2017 than in 2015. This is what we call kind of a species accumulation curve. So the more samples you take, the more times you trawl the net, the more species you'll pick up. And this shows, sort of how many species you're likely to pick up as you take more and more and more samples, and eventually that levels off. Because regardless, if you take more samples, you're not going to find more species. But so you can see that pretty quickly after 20 samples or less, you see this much higher species richness in 2017 than 2015. And those were, we looked at that in more detail, they were mostly Pacific origin species that basically came from the Bering Sea, were vectored up into that region. And that's not unusual. I mean, that has happened always. But we saw this big contrast in 2015, 2017 to show that that's what was happening. And they come with the Pacific water. So the Pacific water was much more extensive in 2017, extended further north, and there was more of it. So this, as some of you may have seen this figure, this is now, instead of the bigger ones on the bottom, this is H0 in the water column. So these are the young of the year Arctic cod in the water column, estimated from hydroacoustics. And so this was the original survey 2012-2013 when we had the Arctic ice cruises up here. And we found, what at the time we thought were very high densities of young Arctic cod in the kind of the Hannah Shoal, south of Hannah Shoal region, basically. Both years here and a little bit over in the Arctic too. And at the time we thought, gosh, these are really high densities, because the densities were as high as like polyp densities in the Southeast Bering Sea. They were pretty small, but there were tons of them, and that's where all the birds go to feed on Arctic cod. And then we went back in 2017, and boom, there were like way more in 2017, and they were all over the place. And 2017 was a particularly warm year. And then 2018-19 got even warmer, but there were not as many. And here is an estimate of the total number, actually, of 2017 just totally stands out, and then it went down again. And that actually kind of fits with a hypothesis that the Canadians have from looking at a bunch of different regions, that as things warm initially, as the production season gets longer, you actually get a better chance of the young Arctic cod surviving, and you get more of them. But, and I'm not showing that here, the fish in 2017 were actually in pretty poor condition. And that could be either because they didn't have enough or the right food, or they were actually out competing each other because there were so many of them that there was like a density dependent effect. So these fish probably didn't survive as well as the ones in 2013. That's the ones that we compared in terms of condition. These were happy and fat, and these were skinny, and the 2013 ones were happy and fat. So they probably survived better than the 2017 year class, but we don't really have a measure of that.
Do the fish school? Are they independent fish? Are they?
The very young ones, they probably school a little bit, but they pretty dispersed. Like if you look at the acoustic images, they usually pretty dispersed throughout the water column or in the top 10 meters when they're small. The adults school more. They definitely are in big schools sometimes. The question for us was, so we had observed this, kind of this density of Arctic cod in that same general region at least, 5 times. And so we were wondering, where do they actually come from? Where do they actually spawn? And so a little bit about their spawning. We don't know much about the spawning, but from some observations in Japan, they typically spawn somewhere under the ice. The eggs themselves are very buoyant, they're very light, so they go right to the surface. They basically rise to the surface. And then they can actually freeze in the ice and they fine. But the eggs are also kind of fragile. They can't handle like turbulence, so they're actually protected by the ice in a sense. Like if otherwise if they were at the surface and there's a storm whipping up the waves, they'd probably get damaged. And then they typically hatch around the time the ice melts, and then the larvae just sort of drift in the water. And to figure out, we haven't observed them spawning anywhere. There have been some observations on the Russian coast. whoever connected, Kathleen, can you mute yourself? Oh, yeah.
They say that they spawn directly into the ice. Seems
That
already
we
close.
honestly don't know. They somewhere in the water column probably, but the eggs, regardless of where they spawn. Actually, I can force mute that. Yeah, I got it. I mute her. They'll spawn somewhere in the water column, and the eggs probably rise straight up to close to the surface or to the surface. So they should be right on the ice, but they've never been sampled. We don't really know. And so my next cruise may be in November or December or January to go look for eggs under the ice if anybody's willing to go. So we were not, we didn't really know how can we, kind of at least get some idea of where they're spawning. And so we actually used a numerical model to do that. So I had a postdoc who developed kind of a, there's an ocean circulation model that kind of tells you where the water currents go, and you can then simulate and basically release simulated larvae in the water column and see where they go. So what we did is we looked at whatever information we could find where they might be spawning, and just sort of based on common sense, and we had seen like larvae, early larvae, and really in the, like small larvae, and we seeded all of these areas, and basically areas up north here, we could exclude those pretty quick. They were just not viable spawning areas. When you release eggs in the models here, they just go off into the basin and they're gone. They're definitely not the source of the young ones that we see here. These ones here were actually intended for saffron cod. So Arctic cod, if we release them here, they basically kind of stay in this area. But this is a saffron cod spawning area. And we did the same thing for saffron cod. And the saffron cod, when we release them here in the model, they end up right where we actually sample them later on. So in the model, we release them and track them until September 1. And then we compare that where we find them on September 1 to what we actually found in the survey, which was centered around September 1. I'm going to have to mute Gay as well. Okay. So, the only viable kind of spawning areas we found for Arctic cod were really the ones on the Russian side, and mostly the ones further south of Bering Strait and Gulf Anadyr, because they were warm enough that the larvae actually had the temperatures that they need to grow to the size that we sampled later on in the summer. So we're pretty sure that a lot of the spawning, that those big aggregations up here, that they originate somewhere pretty far south, most likely even south of Bering Strait.
North Sound didn't have any.
No, Norton Sound was just for saffron cod, and so the Arctic cod, they would just stay in this area. They go, some of them would go along the coast a little ways, but not anywhere close to where we actually sampled. So this probably good. I don't know if you guys sample saffron cod, probably.
Fish from cod is what we call it.
Yeah,
I know it's the wrong one.
Yeah.
He's got the other.
Yeah, and they certainly in Kotzebue Sound too. There's a big fishery for them under the ice, and I don't know if people here fish for them in the winter, but they are a very popular food item. It's just a simulated particle in a computer. So it's a computer model. It's a computer simulation. And so the physical kind of circulation models, they've been around for a while, and they're fairly high resolution, but the resolution is like 3 or 4 kilometers in this area. So it's pretty big areas, and we just release a bunch of particles in the computer in those areas, and then let them drift along, and then we see where they go. And so this actually shows one of the simulations here from all these red areas. So this includes one release at a certain time of year in February, I think, 2012, and where all the particles went from that release. And the key here is that, so they go all over here. So this is mostly from that St. Lawrence release. And there are larvae here in the summer in that area. Some Japanese work that has sampled larvae here, and other people have sampled larvae there. So it's possible that there's some spawning around St. Lawrence. That's why we kind of hypothesize the spawning location there. But they're certainly not the ones that end up there. Almost certainly not, but most of them didn't make it that far. These here, you can't really see that, but Gulf Enadier, they go right along the coast, and the ones that we released here, they go right along the coast, all over the Russian parts, and also over here, and that really is pretty much where we sampled them. So that those were the only releases that actually got the particles to where we actually sampled them in the summer. So I think they probably spawned somewhere down here. There's been some anecdotal reports of spawning.
Mostly like current inside them, or are they migrating?
No, it's currents. These are tiny. They can't swim against the currents. The only thing they can do, and they have some control over where they go that way, they can go up and down. And a lot of fish do that. They take advantage of currents, different currents at different layers in the water column, so that they can go. They can kind of have some control over where they want to go that way. If they work the tides, for example, and fish do that. So our ideas now, this is just kind of a hypothesis that, there are some spawning areas here kind of in the south, and as the ice melts, this is just an example of ice cover that was from January 2018. As the spawning occurs there, as the ice melts, they kind of drift north and eventually end up in the area where we actually sample them. So that's the two surveys that we did in these years. That's where we found the high densities of them. Where they go after that, we really don't know at all. We have no idea. And that's kind of one of the reasons why we wanted to go out in the fall, like we did last November. And so that's.
You assume they go back to where they spawn?
So eventually, that's the key. So, I mean, a lot of fish do this and have long migrations, but these are pretty small. They're not very large, even when they 2 years old or 3 years old. So if they end up somewhere in the Beaufort Sea or along the slope here, they got to get back to this area to spawn. And they have a few months to get there. It's not inconceivable. I mean, some fish in the on the Atlantic side, migrate thousands of kilometers a year to go where they need to go. But it is a long ways. It's a bit, it's 600 miles or so, depending on exactly where they might be coming from. Now, it's possible that there's lots of them over on the Russian side too. They might not have quite as far to go. Now, there have been some surveys on the Russian side. There are definitely Arctic cod here, but it's hard to compare the densities to what we see. They definitely, in the summertime when we sampled here, there's very few adult big Arctic cod. They're not nearly enough to produce the number of young ones. Not even close, not even by an order of magnitude.
Where do they find the Arctic cod?
Well, good question. So we sampled some in the surveys that we did here, but just not very many. On the Canadian side, all the way in the eastern Beaufort Sea, they find them off the slope, off the shelf on the slope, upper to mid slope at like 400 meters down to 1,000 meters in big schools. We've seen that a little bit in this area. There was a NOAA survey in 2008 that sampled some of them up here. And this winter, in November, we actually saw them all along the slope here, not in super high densities, but we had hydroacoustics running, so we did see some along that slope area. That was, one thing we were interested in. Where are they going after we sample them in the summer in the part of the Chukchi Sea? And so that's in November. So we left here on November 6th, and I'm just showing kind of the area that we worked in here. So here's Sikuliaq right there, the northern part of the Chukchi Sea, Hannah Shoal. So this is our track, and we sampled at all of these square boxes. So we got a total of 11 tows, trawls with that big net over 3 weeks. So, we were sharing the boat obviously with others. We had about 6 days for sampling really. The rest was, the other crews are transiting to and from. deployed the net at all of these stations and did a bunch of other sampling, including, so we took water samples to look at the nutrients in the water, chlorophyll and phytoplankton. We looked at like different types of trophic biomarkers, we call them, like stable isotopes that can help you determine what they've been feeding on. And we also took some DNA samples for environmental DNA to see what else is in the water column, like microbes and stuff. And we took samples for Cathy Lefebvre for harmful algal blooms, the toxins that they produce, to check for those. We also had zooplankton nets to take samples of zooplanktons, both to get like density estimates. And also we did some onboard experiments where we actually see, where we were trying to measure whether they actually grow or not, whether they actively growing. And then we had the surface and under ice trawl. That was that net that you see in the water there, that has actually two nets on it. One is a fish net, was a 7mm mesh, and the other one is a plankton net, was a 300 micrometer mesh. And then in addition to that, we had a mid-water trawl and a few samples before we busted it. And then we had the acoustics going the whole time whenever we could. But when you're operating in the ice, a lot of times you have to kind of lower this unit into the water, and you can't do that when there's too much ice around. So we couldn't always run that. And then we also took some ice cores from the ice. So I'll show the video, and then I just have a few more slides after that.
How thick is the ice we're going through?
Where we actually towed the ice was between about 6 and 10 inches mostly, not very thick, so pretty thin ice. Sikuliaq? Well, we were trying to keep the net under the ice, so that was the key. We can't, if it's too thin, it'll pop up through the ice, which is not what you want, because then you scoop up a lot of ice, and we did occasionally. You'll see some of that. We really just wanted to know, do these juvenile Arctic cod, do they actually associate with sea ice at this time of year?
So we're up here to study what's happening in this transition season. And one of the key things that happens from the ocean's perspective is that the surface of the ocean freezes.
I'm Franz Meuter, that's a Beaufort Oceanographer, and you know if you would be able to do it. It's really the Arctic cod. One of the things we wanted to find out. Almost all of these do they associate with the ice starts forming in the fall. The life history in this part of the world we know very little about. That's part of the reason why I was really zero track in the studying Arctic cod here. And we didn't really have a way of sampling them in the winter under the ice. I was aware of the suit, the surface under ice trawl that Outhead originally helped develop to modify to catch fish. And then eventually we have some funding to enhance the fishing capability of the vessel, Sikuliaq. And we were able to have a net custom built for us.
Sikuliaq is quite a new vessel. It was commissioned in 2015. It's a very well-used platform, a very nimble and flexible platform to have scientists come on board and be able to do their work.
The suit is essentially a bottom trawl, but upside down. So instead of being towed across the seafloor, it has floaters on the top that will always make it float on the top.
That's Jim's doing, that was us.
When there's ice above the water, it will glide at the underside of sea ice.
Yeah, the key is really to get it under the ice. That's the difficult part.
Yeah.
But it has a strong tendency to try to stay at the surface.
It is also shearing to the side of the ship in rather undisturbed waters, which helps to sample under the ice, but also in the open ocean samples a portion of the surface water that normally cannot be sampled with plankton or fishing.
We are just above the north slope of Alaska, and our latitude is about 70 degrees north.
About 71 points. The ship is ideally designed for this kind of work because the ice is relatively thin. It's an ice-capable vessel, and they can break through a couple feet of ice at a rate that we still tow the net down. It's kind of ideal for these traditional weight on it, pull it down, and then it goes out to the net.
The ice is freezing very fast. That's the only thing that we have to be aware of. We take too long time to create a thin layer of ice on our runway, and when we fish through that, we will just sample all that ice, and it will be big bag full of ice. The ship was so nice to create us a little runway that you can see here of open water in which we can run the suit up towards the ice. At the end of this runway, the ice makes a sharp hook of 90 degrees. That's what we want. And then hopefully it will smoothly dive under that ice edge, and then we'll be thrown under the ice for another 3/4 of a mile or so. And then there should hopefully be another open patch, according to the radar, where we can take it out. The weather is perfect. It couldn't be any better. The biology is perfect. We have lots of seals and birds around here, which means that there must probably be zooplankton and fish in the water that feed on, and that's what we are after. Sikuliaq are key species of the Arctic marine ecosystem because they are massively abundant, and as such, they are a rich source of the Arctic seal species and a couple of bird species. So they essentially bring the carbon that is created at the base of the food web by algae and zooplankton up to the higher predators.
That's the lights of Prudhoe Bay in the back. So we had to be very adaptable in terms of our sampling. So that's the weight. So that weight weighs 1 ton. Fairly and that really holds developed a little bit of a routine. Deploy the net up the stern like you would any fishing net off a vessel. But the key difference is that the net has attachment points on one side of the frame only. So you can see the pulley that rides on the cable and the weight is attached to that. The water will flow through the net. That flow will help the net shear out to the side. And we hope to do all of that in open water and then into the ice. So then it dives under the ice position. So yeah, that's the net pushing a bunch of ice in front of it, and you hope it dives under instead of eating ice. And it goes pretty smooth under the ice, pretty much everywhere at that point. Easier than the deployment, perhaps. Not always, I'm sure, depending on how heavy the ice is or what you run into.
So we're now going backwards to get into the perfect position to deploy the surface and under ice fall.
I think the dynamics among the scientists and the crew were really exceptional, I would say. But everybody was really respectful, and I think that made for a very positive working environment and for an environment where you can kind of solve problems on the fly. Everybody worked together really well to make it work on this boat. It was kind of remarkable. So those ice chunks are actually, you can not really see that very well. They are shunted out past the net. They don't go into the net, which is the only way you can make it work.
The biggest challenge that we are now here in an autumn situation during freeze up, which means underneath the ice, there's constantly new ice forming, and this new ice consists of microscopic millimeter-sized crystals that are forming in the water and drift slowly upwards. When we fish through this with a suit, we just collect all of them, which is why we very often had our codens full of slush ice, just like a slushy. Yeah.
So yeah, that whole
because this is water very close to the freezing point, about minus 1.9 Celsius. So that has caused us some difficulties, a lot of rinsing of the nets with the fire hose of the ship.
Yep, the fire hose would actually melt the ice fairly gently, keeping the fish and plankton cool. That was not the fire hose. That was just a GoPro in the net looking kind of upward, forward, and some of the ice cliffs look like fish or little organisms coming into the net. It looked like we were catching lots of stuff, but it was mostly ice cliffs.
We have been working really hard with little sleep under harsh conditions, and it was really useful on this expedition that we had a big team. So we could simultaneously process these different size fractions of our catch.
I had very low expectations, and I made that very clear before the cruise. And I told everybody that if we catch a handful of fish, I'd be ecstatic. And we caught in the hundreds. The exciting thing is that we did catch them, because, I mean, from a scientific perspective, whether you catch them or not, it's a result. But if you don't catch any, you never really know, is it because the net's not fishing properly, especially since we were filling up with ice? Are we doing something wrong? Or did we just happen to be in the wrong place? So the fact that we caught them is about the best result that we could expect. What we found was that there is a fair bit of chlorophyll still available, and also zooplankton coming up under the ice that the cod probably feed on. And so we did find Arctic cod that were presumably feeding under the ice. We did take the stomachs out, but at least some of them contained food, so we know they were eating at the time. That is what we hypothesized to begin with. They probably do come up under the ice, and still good to catch them.
For me, it was the same as for Franz. I had low expectations, and I was totally positively surprised to see that we catch them in 11 out of 11 trawls. We had Arctic Cod, so I really hadn't expected that. And I think the idea that they associated with the sea ice at the end of the summer was well substantiated with these data. So that's one of the take-home messages. We also found some surprises from the zooplankton that we have in our catches. There might be a story in that too, because under the ice, we actually found some species that are normally living at the seafloor or above the seafloor in shallow waters. And we found them under the ice in almost 2,500 meter water depth. And this, for me, was a really nice and unexpected results, and something I have not seen in the European Arctic.
And we still have a couple of years to go, really, to sort through all of this and figure out what we actually caught in terms of the zooplankton. The only critters that we have a fairly good handle on right now are the Arctic cod. We counted all of them, but there's a lot of zooplankton that were just put away and preserved in formalin that a colleague will sort through and identify as best as possible. Well, these guys are really good on point research. There were two, a videographer, me had the drone, so that he got a lot of really good drone footage. And they put together a video. He actually worked with an editor on that. So that's, I mean, it's really hard to get a feel for how exactly the net works. You almost kind of have to be there. But yeah, the basic idea is that you have that weight on the cable, so the cable is down at like 5 meters or so at least. So you can then, as it shears out to the side, you can get the cable under the ice, so it doesn't cut into the ice or anything. And you can kind of see that a few times where the cable just went kind of straight down because it had this, the weight itself, the 1 ton weight is in a different line actually, on a separate winch. And so they can adjust the height of that. And so you could lower it down lower to get the cable even lower. Yeah, we went all the way into the basin, so we kind of zigzagged. We found cod all the way out there. In fact, I'll show a couple results pictures that are pretty interesting. So this is a lynx composition. So most of them are probably like late season, young of the years. Although a lot of them are probably H1s because they don't grow a whole lot. But there's a big overlap between big H0s and small H1s. And so we took odorless of everything. So we counted all of them, but we took like every part of every fish is being used for something. We took the otoliths out of all of them. We took stomachs out of all of them. We took livers. We took tissue samples. We were sending whole samples to Louise Koopman and at OSU to look at lipid content. And so there's literally nothing that goes to waste of those fish. So by commercial fishing standards, we weren't really very successful. They laugh at us, but we were pretty excited to catch 170 fish.
And this hasn't been done before. I mean, this net is a new.
Yeah, it's the first time that it's been used in the Pacific Arctic. They did use it in the Eurasian Arctic. It's only been used for catching fish a few times because it used to be, it was developed for catching krill under the ice in Antarctica. That's what they used it for originally. And the guy who built the net for us, was on the boat actually. Michiel van Dorsen, he was here in November and he came on the boat with us too. He's the one who's fished it more than anybody else. And this was actually a lot easier than what they do off the Pelagtan, because the Sikuliaq is so much lower and closer to the water and more maneuverable. We can't bust through 10 feet of ice like the Pelagtan can, but they also broke a lot of frames and nets and things. They've had. So we try to trawl at about 2 knots, 1.5 to 2 knots at least. You don't want to drop below that, because then presumably the fish could potentially escape, although they probably don't swim very fast. Yeah.
2 questions. What's next now that notice that works? And the other is, you said there was chlorophyll, but there was actually reduction in November in the boat.
In November, under the ice was very little light. I mean, there's like not much light. So that's pretty new. I mean, I was not really expecting that, and we only have the fluorescence right now, but we did take water samples everywhere, so we can. Yeah.
Well, people think this is people from afar, nothing's going on under there, but we never like.
Right.
Yeah. So
yeah, that's probably. I mean, these algae are like very adapted to very low light conditions. And the other thing is, like, the major copepod in that region, Calanus copepods, they always start to go down in the winter, just go down deep. And so we weren't supposed to catch them, and we caught them all over. And so that's what we did the respirometry experiments on. They were happy, they were growing, they were feeding. So they don't all hibernate, they don't all go down. Or hibernate is not the right word, whatever they do, diapause things. So just, this is just a map of the catches. So this doesn't really tell you very much because I don't have the depth contours on there yet. We did do hydroacoustics everywhere. The Sikuliaq has 5 frequencies now, so this is more like the zooplankton. There's some layers of zooplankton. This is kind of the fish, and so here at this particular station outside the ice, there wasn't a lot. But as we went across the slope several times, there's some areas with big balls of probably adults down lower. I didn't include that here. We have all the data, we just don't know yet who's going to work it up. Because the license to work up the data is like $35,000. So this was, to me, the most interesting thing. Even though we only had 11 stations, there was a clear indication that it's statistically significant that the density of Arctic cod increases with the thickness of the ice. This is basically a measure of the thickness of the ice. We had sensors in the net that tell us how thick the ice is actually, not technically how thick it is, but the ice draft, which is pretty much proportional to thickness. So the older the ice is, the more Arctic cod there are, and that's, to me, shows that as the ice forms, they probably more and more associate with the ice. The question that we don't know yet is how the relative proportions of what's under the ice and what's in the water column below it, and that's why we had a midwater trawl. But using a midwater trawl in ice is not a very good idea, especially when you borrow a midwater trawl from NOAA and bust it. So we weren't very successful. We got three samples total, and there were definitely Arctic cod in the water column, but we don't have a good handle on sort of the relative densities under the ice and the water.
Was there any change in size? Which size?
Not really, you know, it's very small sample size. We looked at that a little bit, no clear patterns in terms of size. It's not like there were bigger ones offshore, or, yeah. But clearly, I think as we went offshore and the ice became thicker, there were more Arctic cod under the ice. With that few samples, that, you know, it's remarkable that we could tell it. This is just an example of, so we collected macro zooplankton, so those are all the different zooplankton species. And Hauke in the video mentioned a few surprises. And so one of them was this neomyces, a coastal mysid species that really isn't supposed to be there. Well, we found that in pretty high densities over the basin. So both in the water column samples and right on the ice. Whether they just drifted offshore and, you know, just died, I don't really know. We found freestyle stickleback. You probably sample them in the nearshore areas here. You may see them 50 miles offshore, like over the slope. Again, whether they just happened to drift there and were lost, I don't know. Whether they actually make it, I'm not sure. And then what's most exciting to Hauke, because he's a zooplankton guy, were these amphipods that are from this family that live on the bottom in benthos, tube feeding amphipods in coastal environments, and they were under the ice in the basin over 2,000 meters of water, and seem to be happy and doing well. So that's certainly a new observation too. And that's really all. You know, we have lots of work to do, and the samples are just being shipped around right now to different places. A lot of them are going to Germany, and then some here and some in OSU. So that's all I have.
Well, thank you, Franz.
So that's one of Jim Thompson's instruments that he uses to measure the waves in the ice and outside the ice. And this is actually not, I don't know if you used to get pancake ice around you much. Yeah, here you would, because it, you know, you get it when there's waves and you get these pancakes. Up there in the Beaufort Sea, it's relatively rare because you didn't used to have much fetch to create big waves to make the pancake ice, and now, you know, certainly we saw lots of it.
So are you going to, oh, go ahead, no, go.
Oh, these are probably this big. Yeah. The instrument is about this diameter. Yeah. And they can be different sizes, sometimes small, sometimes like this big.
That other yellow thing is the smaller instrument?
That's a float attached to that one. Yeah, and it's got this has a strobe light on it to find it. So what Jim would do, you deploy 5 of those, let them drift for a couple hours. Well, actually not even a couple hours, an hour, pick it back up and bring it back to where they started. And they do that all day long. That's why you saw a spaghetti plot behind where we were standing. That's what they were doing. They were picking up the swift, they call it swift buoys. They pick them up, drop them off, pick them up, drop them off, continuously. So yeah, I mean, you were asking what's next. I mean, I would.
Are we going to see you this fall again?
I'm going on sabbatical next year. I hope to go. Well, I don't know actually if he's going to. But I hope to maybe get some funding to go out. Well, there's a bunch of problems. We think that a lot of them originate on the Russian side. I want to see if we can actually catch either spawning fish or eggs under the ice, and that would be in January, December, January, and ideally on the Russian side. That's maybe the bigger problem.
How big are the eggs?
The eggs,
Like
1.5.
fly fish, like the topeka we eat, or?
They're tiny. They're like 1.5 millimeters, maybe. Very small. Yeah.
I just think it's probably maybe an impossible question to ask, but it seems like in response to changing climate, it's like the total biomass of things, like know that any way to know?
Well, we do that with fish all the time in the Bering Sea, obviously, where we have regular surveys. We just haven't had the It's not an impossible question at all. It's the answer we don't have right now, but
Limited answer for the Northern Bering Sea.
we have some idea in the Bering Sea, as I showed, they basically disappeared. But that's pretty small biomass, really. And that's just sort of the southern extent of the population, I think, and probably not a real big component. They kind of extend into the Bering Sea when conditions are right and then go back. And yeah, we don't really know where the big concentrations are. In the Barents Sea in Norway, the Russians and Norwegians do a joint survey, and they actually have biomass estimates. They do acoustic surveys over the entire region that they distributed, and they have a biomass estimate. They used to have a fishery that it's been closed in recent years because the population has. Yep. Yeah, it was not a very big fishery, but they. In fact, I asked one of the guys one time what they do with them, and he said, "Oh, they fillet, and then we send them to the prisons."
They're good eating.
Yeah. I mean, the fillets are pretty small, but they're a little bigger on the European side. They get a little bigger, but still like small fillets. Anyway, I don't want to keep you longer. It's been over time.
So thank you all. Now, given the situation we find ourselves in with all the new rules for different things, the next Strait Science is scheduled for March 30th. Bob can hear. They're still.
Oh yeah, they're still.
The next one's scheduled for March 30th, and that's going to be Alexis Will, who has for several years been studying seabirds at St. Lawrence Island. So she's going to talk about the seabird die-off. If we have to shut down further, and if she's still on her travel plans, she's not coming here solely to do Strait Science, we will try to do something like this where we'll set it up. If we're not allowed to all meet, then we'll try to have her do this remote. Did this work for those of you who are remote, Bob? Did this come across?
Hey, yeah, thanks, Gay, for asking. Franz, that was excellent. It was good. The only, of course, we had unstable connection for a while, but the video audio was garbled. It wasn't close enough to, I guess, to the speaker. So just, or something happened.
Oh, okay,
Yeah,
for the
yeah.
video portion, but the rest of it was okay?
Yeah, great, awesome.
Okay, good to know.
Yeah, I'll send you actually, or I'll send it to Gay. There's a YouTube version of that video, so you can access it.
See how many.
Awesome.
And then, Bob, did you have any questions for Franz?
No, it was great. Thank you, Franz. Yeah, Vera and I enjoyed it.
Yep, thank you for tuning in.
Thanks for tuning in. We might do it.